everyone. My name is Kim Hobbes and I live in beautiful Conception Bay South, Newfoundland and Labrador. I have a Master of Science degree in biology from Memorial University, and for the last 25 years I've studied the physiology and molecular biology of fish and other aquatic animals. Molecular biology is the study of the structure and function of DNA, RNA, proteins, and other macromolecules essential for life. Molecular biology is used in medicine to diagnose disease and develop new treatments. It's used in forensic science to identify remains and catch criminals, and is used in fisheries and aquaculture science to identify species and to isolate genes that are responsible for growth and reproduction. But before scientists can do any of that, the first thing they have to do is isolate the macromolecule from the cell. Today, I'm going to show you how to extract DNA from strawberries. DNA holds all of the instructions that help with development, growth, and reproduction. They hold the information that makes this strawberry, and makes you, you. The first thing we have to do is get it out of there. For this project, you will need strawberries. You can use fresh or frozen, water, dish detergent, salt, a resealable plastic bag, some measuring spoons, isopropyl alcohol, a coffee filter, and a toothpick or any other piece of wood such as a popsicle stick. That'll be fine as well. The first step in DNA extraction is cell lysis. We need to break the cells apart to get the DNA out. The DNA is contained within the cell nucleus, so we have to break apart both the cell and the cell nucleus to release the DNA. We're going to use both physical disruption and a solution-based lysis to get the DNA out. But let's start with the physical disruption. Physical disruption is applying a physical force to break open a cell. These strawberries are pretty soft, so it's pretty easy to break them apart. In the laboratory, I would probably use some equipment such as a mortar and pestle, or a homogenizer, which is a kind of blender, or a sonicator that uses sound energy to break cells apart. But since we're just using soft strawberries, we can just kind of mush them up. Unfortunately, these strawberries are not very ripe, so I've had to pre-chop a few strawberries to make it easier to mash them up. This is the equivalent of 3 strawberries. So we're just going to put them inside of our resealable plastic bag. Seal it up. And then we're just going to squish the strawberries with our fingers. And you want to try and get an even, mushy mess. Try and break up as many of the cells as you can. If the strawberries were a little riper, or if they were frozen and then thawed, it would be a lot easier to do this. It may take a couple of minutes to get a nice consistency here. This is not too bad. It's a bit fun too, squishing strawberries between your fingers. Okay, that's pretty good. So we physically disrupted the cells now, but some of the cells are still intact even after all that squishing. So we're going to add a solution. It's a lysis solution that is going to chemically lyse the rest of the cells that are not broken down. So let's make our lysis solution. I have half a cup of water, to that, I'm going to add 2 teaspoons of dish detergent. And 1 teaspoon of salt. Just give that a little mix. But try not to create too many bubbles. Now we're going to add 2 tablespoons of our lysis solution to our squashed strawberries here. 1 and 2. Seal up our bag. And just kind of squish this together. Again, you want to try and not make too many bubbles. You just want to kind of get that detergent in, mixed in with everything that's in there. So what's the detergent doing? If you think about when you wash your dishes and there is grease or oil or fat on your dishes, the detergent breaks those fats apart and allows you to get your dishes clean. Similar principles happening here. The detergent is breaking up the lipids that are part of the cell membrane, and that allows the cell to break open and release its contents. So basically what we have here now is a mixture of water-soluble contents from the cell, and that includes our DNA. And we also have some water-insoluble components such as the cell wall and the seeds that were on the outside of the strawberry. So next thing we have to do is separate our soluble components from our insoluble ones, and we're going to do that by filtration using our coffee filter. So we're just going to pour our mixture into our filter. And we're going to let the liquid portion filter through. You can squeeze the filter a little bit, just be careful not to break it. Like I just did. Oh dear. Okay, so that didn't work so well. So we'll get rid of the paper filter. You know, no scientist ever got anything right the first time, trust me. So we're going to use a different kind of filter now. I just have a basic sieve here, and we'll just use that. Try again. Ah, that's much better. I'm just going to use this spoon to kind of push some more of the liquid out. I feel like I'm making jam. Okay, that's pretty good, so we'll go with that. So what we have here now are all of the soluble components that was inside the cell. So we have a mixture of DNA, RNA, proteins, enzymes, all sorts of things that are all water soluble in this cup. But we still need to get our DNA out of solution if we're going to do anything with it. So we have to precipitate it. And that's our next step. DNA has a negative charge and water has a positive charge. The water interacts with the DNA and keeps it in solution. We need to get it out of solution, so we have to somehow break that bond. Remember the salt that we put in the lysis solution? The sodium ions in the salt is sodium chloride, has a stronger positive charge than the water. So it's going to bind to the DNA and neutralize it. And it'll help it get away from the water and come out of solution. But the bond between the sodium ions and the DNA is not a really strong bond. So we need to try to strengthen that bond so we can pull the DNA out of solution. And we're going to do this by adding an alcohol. And in our case, we're going to use isopropyl alcohol. You're going to want to add an equal amount of alcohol to the volume of water that you have. And we're going to carefully go down the side of the cup because you want to try not to really mix the alcohol and the water layer. You want to try and keep them separate. That looks pretty good. So the alcohol is a separate layer from the water here. And as the alcohol molecules start to replace the water molecules in the solution, that bond between the sodium ion and the DNA is going to become stronger, and the DNA is actually going to start to come away from the water. And if you look closely, you can see a cloudy substance starting to appear in the alcohol layer here and in between the alcohol and water layer. See the white stringy bits? This is the DNA precipitating out of solution. And if we take our toothpick now, we should be able to hook it out. Here we go. This is DNA. Now, if I were in the lab, I'd need to clean this up a bit before I could use it because some of the proteins that were in solution and the salt has also precipitated out with the DNA. But as you can see, that's quite a bit of DNA just from 3 strawberries. And the reason for that is strawberries are octoploid. This means that in every cell, they have 8 copies of all of their DNA. Most other organisms, including ourselves, only have 2 copies of DNA, one from our mother and one from our father. I hope you enjoyed this video, and I hope that you will try this experiment at home. If you want to learn more about me, you can check my page on the WRDC website. I encourage you to try this at home. Try using different kinds of fruit or other types of food and compare the amount of DNA that you can extract. Thanks for watching.